Hi, my name is Sangeeta Bhatia, and today I'm going to be telling you about tissue engineering of the liver. To orient you, this is a schematic of the liver. The liver has over 100 billion cells and performs over 500 functions in your body. 300 million people worldwide suffer from liver disease, which one can acquire from viral insult, like hepatitis, or a drug insult, like chronic alcohol exposure, or even an over-the-counter drug in high doses. What happens when the liver gets damaged is it forms scar tissue, seen here in the middle panel. The scar tissue evolves into this nodular pattern known as cirrhosis. A cirrhotic liver is prone to developing cancer over time. So patients who have liver disease often progress to liver failure and liver cancer. The gold standard for treatment of these patients is whole organ transplantation. However, there are not enough donor organs available for transplanting those in need. Because of this, and because the liver provides so many functions that are vital for life, many groups have thought about supporting liver function with cell-based therapies, wherein the cells would be hepatocytes, the cell of the liver that performs most of those 500 functions. The idea is that living cells would perform liver functions in a variety of hybrid formats. So here I'll just show you two examples. These are extracorporeal devices. They are akin to kidney dialysis machines. The blood would run outside of the body through this cartridge, and the cartridge would house living hepatocytes that would process the patient's blood. The idea is that this device would bridge the patient to transplantation or even support the patient while the liver regenerates. Because interestingly, the liver is one of the few tissues that actually can regenerate in certain kinds of injury. Another idea, which our group has been pursuing as well as several others, is this idea of an implantable tissue engineered liver. This would one day, we hope, serve as an adjunct or an alternative to liver transplantation. The problem in the field has been that liver cells in these devices do not perform the 500 some odd liver functions that are necessary for patient support. We know from a variety of work in the field that this is because the liver microenvironment depicted here is disrupted when the cells are isolated and put in contact with the plastic of the dialysis cartridge or the polymer scaffolds that are going to be implanted in people. So the liver microenvironment, as you can see, is quite complex. Here we see a repeating unit of the liver. This is called the liver acinus, and each of these units is about 1 millimeter in diameter. The hepatocytes, the cell of interest that performs most of those 500 functions, are aligned in these cord-like structures. These are actually three-dimensional structures known as hepatic plates. And you can see that along each one of these cords lies a blood vessel. So each hepatocyte is only 1 or 2 cell widths from the blood flow, and that provides them the ability to very efficiently process the blood, both to metabolize drugs and hormones in the body, as well as to produce secreted products for the bloodstream. For example, your liver makes most of the clotting proteins important for clotting blood after you cut your finger. In addition, it makes albumin, which helps you keep your fluid inside your blood vessels. It metabolizes all the drugs that you take from your doctor. And in addition, it regulates energy, it regulates energy metabolism. So all of these functions are important, and they reside in these hepatocytes that are dependent on this very complex microenvironment to function. When we remove hepatocytes in order to incorporate them into one of these cell-based therapies and put them in culture, this is what happens to their function. So here I'm showing you data for just one liver-specific function, and that's the production of albumin, which is important for oncotic pressure, the fluid pressure in your bloodstream. And you can see that these are hepatocytes here. They've been cultured on collagen-coated tissue culture plastic, and after a couple of weeks in culture, they're spread out and they're dying, and all of their liver-specific functions dramatically decline. So in our group, what we've been interested in is seeing whether we could recreate a happy microenvironment for these hepatocytes outside of their native microenvironment in such a way that it could be useful for incorporation into a therapeutic device. Because the environment of the cells in vivo is specified at the 10 to 100 micron length scale, we have been drawn to technologies that allow us to manipulate the 10 to 100 micron length scale. And in fact, if one looks at the computer revolution was driven by miniaturization technologies that have exactly this property. So what I'm showing you here is what we achieved as a community over 50 years of progress from a single transistor to 100 million transistors through a very simple process known as photolithography. So this is a process by which computer chips are made. And what's done is one takes crystalline silicon, coats it with a light-sensitive material, shines light through that light-sensitive material, and creates a pattern on the surface. Now, if you were making a computer chip, one could use that pattern then to make integrated circuits, resistors, transistors, and so on. But we and others have been thinking about using these technologies to create cellular microenvironments for the applications of tissue engineering that I mentioned. In this talk, what I'll be telling you about is our work in constructing in human tissue microenvironments for the liver, our interest in interrogating these microenvironments in high throughput for example, drug development, and furthermore, in our interest in using these liver tissues for studying the interaction with pathogens that only normally interact with the human host. So I'm going to start by telling you how we're interested in exploiting these tiny technologies for constructing engineered liver tissues. So the first step that I need to tell you about is how we adapted that same process I mentioned earlier, which is called photolithography, where normally one patterns metals on silicon, here to pattern cells on glass. And what we did was we took the same liver cells that I showed you before, now they're colored red, and we've surrounded them with another cell type. So we knew in the liver, in its native microenvironment, there's at least 4 other cell types that are close to the hepatocytes. And so here we've just added one other cell type back, and this is a randomly organized co-culture, a co-culture of 2 different cell types. What we did with photolithographic processing was create what we call a micro-patterned co-culture. And that's shown here. In this system, what we've done is use the same light-based systems to pattern collagen extracellular matrix on a glass cover slip. The hepatocytes, the liver cells of interest, stick on those spots, and then we can fill in the spaces with the supportive cells. And in this way, we can control the architecture of the tissue in a 2-dimensional plane. And what we found over the years is that the geometry of this architecture, the control over which cellular neighbors are right next to the hepatocyte, whether they're self, homotypic, or non-self, heterotypic, determines the level of function that one gets. And in particular, for human tissues, that one can rescue the phenotype of the cells, those 500 functions of interest, for about 4 to 6 weeks. In addition, we've been interested in using another version of microfabrication technology known as MEMS. In this process, instead of just making flat, planar pattern structure, what groups have done over the years is develop etching techniques to etch crystalline silicon, like this part that you see here. So this part is first patterned with light, and then deep reactive ion etching is used to etch out the structure that you see there. Using this part, we were able to manipulate our cells at the microscale. So here what we've done, you're looking at 2 interlocking combs. You see one cell population on one part of the part and the supportive cell population on the other part. And what we're able to do is move them together and apart and ask questions about whether they need to signal each other continuously or reciprocally. So for example, in this experiment, in this movie, what's being done is the cells are being brought together using a simple micropipette manipulation in a biosafety hood. And you can see that they've been brought together in what we call gap mode, where they're separated by an 80 micron gap, but they're not allowed to touch. Now they've been brought into contact mode, where they're allowed to touch over in contact mode, and now they're separated completely. This allows us to study the dynamics of cell-cell interaction using microfabrication. What we've learned doing as an ensemble in these set of experiments is the following: that this big fat cell here is supposed to represent the hepatocyte, and this cell is the supportive stromal cell. In this case, it's a fibroblast. What we've learned is that the cells need to touch each other for about a day in order to get this rescue that I described earlier. After the first day, however, they can produce soluble factors that can diffuse over that 80 micron mode. And those soluble factors are completely sufficient to support the hepatocyte. So what's exciting to us about this finding as engineers is it might suggest that after initial priming period, one could support hepatocytes in an implantable tissue or in an artificial liver device without needing to have another living cell taking up space and nutrients. Okay, so I've told you about how we used microfabrication to learn about how to stabilize the liver cells, but one can also use these light-based patterning techniques to build three-dimensional implantable parts. And in doing this, we were inspired by this rapid prototyping technique known as stereolithography. So stereolithography is a technique whereby one will draw a three-dimensional part using CAD software in the computer, and then use it to drive this robot. And the robot works as follows. It drives this stage. The stage can move up or down in a vat of light-sensitive polymer. So when the stage is at the top and one shines a light in a pattern, it cross-links the polymer, and then we can make it drop a level and shine a different pattern, and so on and so forth. And at the end, you build very complicated three-dimensional parts. And in fact, there are programs now to be able to drive these with three-dimensional patient-specific anatomic data. So one can imagine then taking CAT scan data and driving a robot like this to make a three-dimensional living part. So in order to make a living tissue using a system like this, we need to create cells within this three-dimensional light-sensitive system. In order to do that, we borrowed a chemistry that was first developed by a scientist named Jeff Hubble, which is described here. This is a polyethylene glycol polymer. So it's a long chain of polyethylene glycol molecules. It's a long chain polymer with reactive end groups. And what one does is simply mix in a photoinitiator, a light-sensitive chemical, with cells. And when one does this mixing and shines light, you get a photo-cross-linked network with cells embedded in the polymer structure. So this is like the cells are fruit and the polymer is the jello. So the cells are the fruit in the jello. And the nice thing about this is that one can then change the pattern of light that one shines and create different structures. So in order to get hepatocytes, liver cells, living in this system, as I mentioned, they're very sensitive to their microenvironment. A graduate student in our group spent about 5 years discovering how to do that. And in this slide, I'll summarize her work. So what she showed was that if you put liver cells just in this material system and tracked the albumin secretion, which is one of those liver-specific functions I mentioned earlier, they survive, but they don't function like liver cells. If instead you add those supportive cells that I showed earlier, the cells do a little bit better. If one further adds a peptide into this inert polymer, this is an RGD peptide, which mimics the binding site of fibronectin, an extracellular matrix molecule in the liver, as it interacts with the integrins, the receptors on the hepatocytes, one gets even more function. And finally, if one adds a third cell type which exists in the liver, the endothelial cells, which are the natural neighbors to the hepatocytes and normally live only 1 micron away from the hepatocyte, if one adds those into the network, now one can get a highly functional liver cell in this photo-encapsulated system. So using this system, we've created many, many different PEG-based tissues. And this is just a smattering of the kinds of architectures we've been able to make over the years. In this movie, what you'll see is in this latest version, we're now able to make these microfluidically. So this is another type of microfabrication where one can make a microfluidic device out of polymers. Here the polymer is called PDMS, polydimethylsiloxane, And what you see entering into this microfluidic nozzle is a mixture of the cells and the polymer suspension. As they bud out into these droplets, then they are ready to have light shown on them, and then they can be polymerized in these microfluidic droplets. And this is an example of cells inside these microtissues. So we've made progress now on the fabrication of these hepatocyte-based tissues. The next thing we wanted to do was see how they would do in vivo. And that experiment is shown here. So in this experiment, what we've done is taken these hepatocytes, these liver cells, and genetically modified them with a virus, a lentivirus, that drives a gene known as the luciferase gene when a particular promoter is active, the albumin promoter that I was mentioning earlier. So if the hepatocytes are happy and functional, they will express luciferase. And here what we've done is implant these tissues in the intraperitoneal space, the abdomen of the animal, or in the subcutaneous space. So we're looking at different sites of implantation. And then we look for light generation by this tissue as a marker of how it's doing over time. You can see in these experiments, these tissues survive in the intraperitoneal space for up to 3 months by this measure. And if we look in the blood of the animals and look for markers of the human proteins in the mice, we see that the cells are making human albumin in the blood of the animal. And this is a sign that the blood vessels in the mice have been recruited to that construct and they're hooked up. So the construct has been vascularized. Okay, so that's as far as we've gotten in terms of fabricating tissues that we hope to implant in patients one day. We have lots of work to do on the scale-up of these tissues. The tissues that I mentioned to you only have about 1 million cells in them, and in order to get a therapeutic effect in patients, we probably need 1 billion cells. So we have lots of ideas now about how to scale up by 3 orders of magnitude. Hopefully next time I talk to you, I'll be telling you about that. What I'd like to now mention in the last 2 parts of my talk is how you can use these model systems not just for therapeutic applications, but for scientific discovery by interrogating them. So the application that we first became interested in when we realized that we had human microtissues growing in the laboratory was this one. This is the infamous drug discovery pipeline. And what you can see here is that typically one can start with about 10,000 chemicals, and over the course of about 15 years and close to $1 billion, one can get one FDA-approved drug on the market. What's problematic is that actually even after all of the preclinical studies that are done, all of the in vitro screens and the required animal screens, so these are typically rodent models and large animal models, when one enters into phase 1 trials, which is when the drug is first exposed to patients, 1/3 of the time they exhibit liver-specific toxicity. So they exhibit toxicity to the human liver, which was not predicted earlier in the development process. So we were interested in this idea, which was whether we could bridge the gap between animal models and clinical trials with some of these technologies. We could make engineered human livers in vitro and use them to study this process. So what I'm going to tell you about now is a 2-dimensional in vitro human microliver. And one can also do the implantable 3-dimensional models that I mentioned earlier to humanize a mouse model and, again, bridge the gap between mice and clinical trials. So when one discovers new drugs, one often doesn't have very much of the compound. So you'd like to develop a much higher throughput way of fabricating the tissues than the ways I've mentioned in the past. The photolithographic process that I described to you in the beginning of my talk was one wherein we patterned cover slip by cover slip, and we created these microarchitectures that would stabilize the cells for about 4 to 6 weeks. But that's not a practical way to screen thousands of compounds. So one thing that our group worked on was miniaturizing that technology further. We came up with this. This is a multi-well device, and the idea is that this looks just like a 24-well or a 96-well plate. This is sort of the workhorse of the field of biology, and this can be fed with fluidic robots. At the bottom of each of these wells is a micropatterned co-culture of the two cell types that I mentioned earlier, human hepatocytes and the supportive fibroblasts. And now they're organized in their optimal size and shape for human cells. The way we do this is we create a soft polymer part that has a stencil at the bottom of each well. When you pour collagen through this stencil, you get collagen spots on the underlying surface. And now, again, as before, you can seed human hepatocytes and then surround them with the stromal neighbors. And in this way, one can reuse this part over and over again and never have to go back to the microfabrication facility and have a sort of high throughput format of the microliver tissues. So if one now looks at the albumin function of these tissues, and this is the curve I showed you earlier where the albumin function is lost very rapidly, one can see that it's rescued and it's stable for about 4 to 6 weeks. So the next question then is, are these useful for drug screening? So one important aspect of liver tissue in vivo and in vitro is the one I already mentioned, which is that animal livers and human livers differ in their drug metabolism. There are a couple of reasons for that. The first are a class of enzymes known as the P450 enzymes, and they have species-specific differences. Furthermore, there's a process known as induction. Induction is the process whereby exposure to a chemical upregulates the P450 metabolism and alters the metabolism of that drug. So the induction process also varies in a species-specific way. So what we wanted to do is make micropattern co-cultures out of rat cells and human cells and expose them to inducers that are known to have species-specific differences and ask whether in vitro we could predict these findings that are normally reported at the level of the organism. And you can see here with these two drugs, omeprazole and beta-naptoflavone, we were able to upregulate the P450 metabolism in the human system and relatively less in the rat system, and that recapitulates what we know about the clinical picture. So this was encouraging for the utility for species-specific induction studies. Furthermore, this process of induction is actually the mechanism by which drug-drug interaction occurs. So many of you may have noticed when you go to the pharmacy and you get prescribed a medicine, it'll tell you sometimes not to take the medicine with another drug, that the two drugs might interact. And the drug-drug interaction often happens at the site of the liver. So this is an example of Tylenol, acetaminophen, exposure to the liver. And so what you can see here is that the acetaminophen exposure to the liver is not consequential. These two drugs, one which is an anti-seizure medicine and the other which is a heart medicine, are not consequential to the liver. However, if you give acetaminophen together with the anti-seizure medicine or with the other one, then you see the emergence of drug-drug interaction. And we know the mechanism of this has been well worked out, that that's recapitulated in this system. So this is promising for the ability to maybe study drug-drug interaction in humans, again, in vitro and high throughput. And finally, one can then study the toxic interaction of the hepatotoxicity of drugs with these livers. So this is an experiment where one calculates the toxic concentration at which 50% of the cells die. That's called the TC50. And one can do this over 24 hours of exposure with a variety of clinical compounds. And this is what's typically done now for novel small molecules. And one can rank order these chemicals, and we can see that they rank order in the way that they should based on what we know about clinical exposure. What we're more excited about is the potential to do things actually differently than the way people do things now, and that's to do chronic exposure. We know that all of us take our medicines in low-dose, multi-day format. And if one looks at the emergence of hepatotoxicity clinically in clinical trials, it actually usually unfolds on the order of weeks. So in similar experiments, what we've done is shown that one can do multi-day exposure and score the relative toxicity of compounds over a chronic lifetime And that, in fact, the answers change if you do a chronic exposure as compared to an acute exposure. So we're excited about the potential to change the way people do toxicity testing with small molecules. Okay, so I've told you about building implantable livers, about building arrays of tiny livers in vitro. And the last thing I'd like to tell you about is how one might learn something about pathogens that infect the normal human liver using a platform like this. So in fact, I've already told you about one class of pathogens that infect the human liver, hepatitis. So there's hepatitis A, B, C, D, and many more. And in particular, we've been working on hepatitis C, for which there's no vaccine and no good effective drug therapy. Another pathogen that's very interesting, for which there are not good in vitro models, are the human malarias. So just to remind you how the human malarias work, Plasmodium falciparum and Plasmodium vivax are two parasites that are transmitted by a mosquito bite. So typically the mosquito will bite a human. The sporozoite, the parasite, will travel through the bloodstream, infect the liver. When it infects the liver, it will traverse several hepatocytes and pick one in which to set up shop. It sets up shop in that hepatocyte. It will then multiply and grow and undergo morphogenesis, and then burst out into the bloodstream, where it can then infect the erythrocytes, the red blood cells. And it's at this stage that one would get the clinical symptoms that one associates with fever. What's interesting and exciting about the opportunity to get drugs to work on the liver stage is that typically 30 sporozoites going into the liver would be amplified to about 300,000 merozoites bursting into the blood. So if one could kill the parasite at the liver stage of infection before there were any symptoms, one could prevent the symptomatology and also this amplification in the patient. So the trouble with studying all of this is that the human malarias don't infect animal models. So, for example, they don't infect mice or rats in the laboratory. So we've been interested in studying our human microlivers in vitro and seeing whether one can infect them with the human malarias and recapitulate this liver stage of the malaria life cycle, both for drug discovery of new antimalarials and for vaccine applications. So in this movie, what you see is a typical sporozoite that would be swimming through the bloodstream. And you can see what they do when they swim like this is they undergo this gliding mechanism. And as I mentioned, they traverse several hepatocytes on the way to picking the one in which they set up shop. The hepatocytes that they traverse on the way are wounded. So the membranes are temporarily breached, and then they self-seal if they survive. So we've recently been infecting our microlivers, those 500 micron colonies of hepatocytes that I mentioned earlier, with the human malarias. So here what we've done is add a fluorescent dextran, a red dye, to the media. And as the sporozoite traverses through the cells, it injures the membranes, and the cell take up the dye, and if the membranes seal, then they stay lit up like this. And so essentially this is a molecular footprint of the sporozoite traveling through our human microlivers in culture. And so we've now done this with both Plasmodium falciparum and Plasmodium vivax, and we're very excited to get a glimpse of the elusive stage of Plasmodium vivax that's referred to as the hypnozoite. It's a dormant stage of the parasite that no one has ever seen before in vitro, and we're dying to see if we have it growing. So just to summarize, I think that the tiny technologies are very powerful for the study of tissue engineering and tissue microenvironments. And I've given you the example of the liver, but I think this is broadly true because the microarchitecture of all tissues actually have repeating units on these same length scales. So here are just a list of technologies that are emerging in the bioengineering field from the 100 micron length scale all the way down to the 100 nanometer length scale that are really ripe to be borrowed in this field. Just to summarize then, I've told you how one can use microscale technologies to fabricate liver tissues, to interrogate arrays of liver tissues, and then finally to learn more about pathogens that infect the liver. So thank you for watching, and I'd like to thank our group for all the hard work and in particular Megan Shan and Alice Chen, who helped me work on this talk.